Hi everyone, welcome again to the AWM Pitt Grad Seminar. Our next speaker is Anna Thomas. She is a PhD student from University of Pittsburgh. Her topic today is on computational model investigating the role of rodent substantia nigra pars reticulata projections to pedunculopontine nucleus in long-term motor recovery in Parkinsonian rodents. Anna, you could go ahead.
Thank you very much. Let me pull up my slides. All right, can everyone see the slides?
Yes.
Right, there we go. All right, I simplified the title a little bit. Sorry, I should have updated you with it, but hopefully this one will be slightly easier to understand. Yeah, thanks Farjana for organizing this. I'll be talking about the project that I'm currently working on, and since I think the background of people here is not quite math neuro, I'll try to take a few minutes to explain a little about the background of math modeling of neurons. So this is work with my advisor, John Rubin, and we collaborate with Erin Giddis and her lab postdocs, as well as some students who are performing the recordings over at Carnegie Mellon. So the title is computation. We're building a computational model of the basal ganglia projections, which are the collection of nuclei we see here on the bottom right, and specifically the output nucleus, which is this orange one, to the brainstem, which is a little bit further down here. And specifically, we're looking at rodents in a Parkinsonian state, so we kind of put them artificially into a disease state, and in response to some stimulation, they see some very long-term motor recovery. So we are currently building a model to hopefully eventually explain some of these phenomena. So a few words that will come up many times today are the SNR, which is this last output nucleus of the collection within the basal ganglia, this little orange bit, and then the pedunculopontine nucleus, the PPN, which is just a population of neurons, a set of neurons within the brainstem. So a little bit of background on modeling a neuron. If you've seen a model neuron or a sketch of a neuron before, this is probably what you've seen before. And a lot of what goes on in the brain, a lot of the signals that are passed on between billions and billions of neurons are electrical signals that propagate from one end of the neuron to the other. So one thing to note is that neurons can receive inputs from many other neurons and they can project to many other neurons, but for the most part, rule of thumb is that it just travels in one direction. So signal just propagates, as we see here by the arrow, along the axon of the neuron. And if you've heard of an action potential before, so the membrane potential is the difference in voltage between the intracellular or the inside of the cell of the neuron and the extracellular fluid, which tends to be around -65 in general. And if we see a sharp increase in the membrane potential and then followed by a rapid decrease and some maybe overshoot over here, that's called an action potential. And it's considered to be an all or nothing process. So either a neuron exhibits an action potential or not. There's no like half action potentials or signals that could maybe be confused or need some kind of threshold to be determined. And the next question is, how does a neuron change its voltage, or how does this change along the neuron? So there are many different ions on the inside and the outside of the cell. Sodium and potassium are very common ones. Sodium, for example, is more concentrated in the fluid around the neuron than on the inside. And since the membrane voltage on the inside is more negative than outside, if a channel were to open that lets specifically sodium ions in, then the sodium would want to flood into the neuron. And that would then result in the membrane potential becoming less negative because sodium is a positively charged ion. And neurons do in fact have these channels that specifically let only through single types of ions, and we have a similar thing for potassium. And since the potassium concentration is greater on the inside than on the outside of the cell, if a channel were to open and the membrane potential, for example, at this point is very high, then potassium ions would want to flow out of the cell. So since these channels, and there are many along the neuron, open at different rates, that's what results in this kind of action potential shape. So sodium opens faster and makes the membrane potential more positive, but then sodium shuts off and, for example, potassium opens, which then polarizes the cell again and drags it back down. So that's the general idea, but there are many different types of ions and ion channels. But this is the general idea. So we have a total current is the sum of the membrane capacitance times the potential change across the membrane. So this will be our ODE in the end. We have the membrane voltage change with respect to time. And then we also sum, for example, this is the potassium current, where G is the conductance. N in this case is also an opening probability, just like M. There is no inactivation, no closing probability of the channel for potassium. And then we just have the membrane voltage minus some constant. And these are, the next one would be for sodium, a very similar formulation, but within this M and H, there are time constants that help determine or help create these action potential shapes. And these are just three basic currents. We will see later in the model that we're making, we have added many more to create more rich dynamics. What else do I want to say? Yeah, so this is a standard, pretty basic model, kind of like the starting point. And one more thing I want to say. So this is an ordinary differential equation. No spatial properties are considered here because we consider we basically neglect or consider negligible the time it takes for this action potential to travel across the neuron. So we're just modeling them as points where all the channels are basically just at that point where the neuron is. And when we connect neurons, that's when they might have some external input or like an applied current. So the motivation that got us started with this project was that a paper was published a few years ago by Aaron Giddis and some more people, where a very specific subset of neurons were stimulated. So just basically saying nobody else was affected except for some very specific population within the basal ganglia. So this is striatum, and here that SNR population I talked about earlier. And after stimulating it during these blue pulses, so over the course of 30 minutes, 10 stimulations, rodents, which were previously immobile because they were artificially put into a Parkinsonian state where the symptoms are usually a very difficulty to onset motion, so they're mostly immobile, mostly stationary. And after this repeated pulsing, they were able to move much more than before. So they were seen running around, and under some circumstances, even they were seen running around for many hours after the stimulation was over. So this is analogous to, or I guess this is under the greater category of what is performed in deep brain stimulation in humans with Parkinson's disease. So there's some kind of stimulation going on, and then ideally after that, there is some motor recovery or symptom relief. So the question is now, or on the left here, we see the non-diseased state of these gray boxes, which are mostly nuclei in the basal ganglia, and then the one that is in the brainstem is this PPN. So currently, many models exist of these different nuclei in the basal ganglia, including the SNR, which my advisor has worked on, as well as the postdoc John Parker. He is working on a model that I will use a part of in the future. But our job is now to try to explain how this plasticity, which is kind of like long-term changes to the system, are possible through the stimulation. And what is known about, or the models that exist for these upstream nuclei, do not currently suffice in explaining what is going on here. And since the output nucleus, so everything that goes on in the basal ganglia, the next step, or where the signal propagates to next, is the brainstem, when on its way to, let's say, like muscles and actual motor output, would be the brainstem. So the experimentalists have looked at PPN and identified that the neurotransmitter, or some of the properties there, could be helpful in explaining this kind of plasticity. And I forgot to say, these arrows are just how the nuclei are connected. We can see it's fairly complicated. The black arrows mean that the source where the arrow is coming from, when it's more active, the end of the arrow, the receiving part becomes less active. The gray ones mean that that's the opposite. If this one becomes more active, then the receiver also becomes more active. So these have been studied quite a bit, and we now want to model these two populations specifically and use what we know about the connectivity to explain some of the dynamics. We see they're also indirectly connected through other loops that close back to SNR. And on the right here, we see just a depiction of what might be changed or what is commonly different in the Parkinsonian state. We see that some of the connections are strengthened, some of them are weakened. So this is kind of the diseased, slightly dysfunctional state. So if we get to the model that we're working on right now, we see some terms that popped up in the earlier model slide of a neuron. The sodium current is the same. Sodium, potassium, and the leak channel are just as before. But neurons exhibit many, many different types of ion channels, and it's basically our job to figure out which ones do these PPN neurons have, what equations describe them very well, or the dynamics of it. And also it's always a balance of between how much detail to include, but also what is maybe computationally feasible over the time frame that we're trying to simulate, and also which ones contribute majorly to the dynamics. So the sodium, potassium, and leak channel have the same formulations as before with some probability of activation, and this power depends on how many processes are involved in, for example, opening a channel or closing a channel. And then some of the interesting things about these three different types of calcium channels that we have included here are that some of them are not just voltage dependent. So for many of the channels, the opening and closing of the channel depends on voltage, just the current membrane voltage. But for some of them, they only depend on the calcium concentration on the intracellular side, or some have, in this case, a mix of voltage as well as calcium-gated currents. And they have different time constants, so they all operate at different rates. And one of the tricky things here was that this model for PPN, this kind of ODE model of this formulation does not quite exist. So it was our job to read up on the literature and to figure out which currents are exhibited, and then to find out how have these currents been represented in papers in the past. And there's not quite a consensus on them yet, but we've chosen the ones that we think are plausible and would contribute to the dynamics that we're trying to recreate. And one of the differences, or one of the things that will give us some wiggle room in the parameters, is that some were, these currents are from models, for example, for different animals, or for if they're maybe sliced. So if a slice of an animal brain was taken and then measured, then there's also different connectivity and different temperatures, many different factors that play a role in these. So those will allow us to tune our model. And one good benchmark to have when building a model is a frequency current, an FI plot. So this last term that I haven't mentioned yet is we can either in experiments apply with an electrode a current to a neuron and see how it fires in response. So if and how frequently action potentials are exhibited by this model or by the neuron. So this is this term here, or it could also be some input from a different part of the brain or different neighboring neurons. So with what magnitude of current does the neuron or the model fire at what frequency? So these are experimental recordings. And we can see here that, for example, in the last part of this figure, for an applied current of 200, 250 picoampere, the neuron fires very, very rapidly. So these are action potentials, each one of these vertical lines, but even these smaller ones, each of these is an action potential as well. So the interesting thing about these neurons is that even for very, very large applied current, they still continue to fire and fire more rapidly, or as it plateaus a little bit here, as we can see in the frequency, versus for other neurons, it's more commonly seen that once the current is too large, that the timescales of the constant, they cannot either pull the voltage back down fast enough or not activate fast enough, that they just go quiet once the applied current is too large. So that's definitely something we'll be looking to match in our model, and that's something we're working on right now. And one other thing I wanted to point out here, at zero current, the curve is not quite at zero. It's maybe between 1, 2, 3 hertz. So that's also something interesting to consider, that when there's no applied current, the neuron does on average fire maybe just a few hertz per second, a few times per second. So this is an actual graph of the model right now, and I've included the same plot, this FI plot from the last page. On the y-axis, we have the membrane voltage, and on the x-axis, time in milliseconds. So these action potentials happen very, very rapidly. They're usually on the timescale of just a few milliseconds. Or in this case, in this top panel, there's no applied current, and we still see oscillations with the parameters as they are right now. So that's a good starting point at least. And now I want to show just an example of what some of the currents look like. So this black curve is still the membrane voltage, but now we have, for example, the potassium current in red, which is positively charged ions leaving the cell, versus the green plot, for example, is a calcium current. So those are positively charged ions entering the cell. That's why they have opposite signs. But we can see that they have different timescales. These two activate roughly at the same time, versus the purple one takes a little bit longer. So those are some of the intricacies of these channels. And when tuned differently, when we change some of the conductances, some very interesting things can happen. And so one of the next steps that we could do in analyzing our model and determining some of the characteristics about it, maybe depending on a parameter. So if we vary a parameter, what would change about the firing properties? How could we get the oscillatory period to be very, very long compared to the other neurons where we want them to shut off for a sufficiently high current? So that's where bifurcation diagrams become very helpful. So if you've taken an ODE course before, this might sound familiar. So they're helpful in finding different parameter regimes for maybe the resting state where the neuron is not able to fire. Maybe if the current is negative, for example, then the voltage of the membrane is very, very low and the neuron does not fire at any point. Or if we crank it up a little bit, we see these oscillatory states and then maybe their stability as well. So this is a sample bifurcation diagram from a different model. It's just the basic sodium, potassium, and leak channel. So it's not quite ours yet, but I wanted to show what we would maybe want to do in our next step, since we're still fairly early on in the project. But here in red, we see a stable, steady state. So for this amount, on the x-axis, we see the applied current, and on the y-axis, the membrane voltage. For some applied current within this red region, we would never see oscillations of the model. But if we start to bring the current up into the region where these green lines are, then the steady state would, the state where the neuron or the model does not fire becomes unstable, and actually the model wants to fire or wants to oscillate. So while this plot does not say anything about the frequency, since we just have voltage and current, we do see something about the membrane voltage or the amplitude of the current. And we can see as the current becomes larger, the amplitude becomes smaller. So that kind of agrees with what we saw in the benchmark plot. And here, where there is a transition from these stable oscillations to the stable steady state again, so for too large applied current, the model will not fire anymore. This transition, this change in quality of the solutions or numerical approximation to the solutions is called an Andronov-Hopf bifurcation. So if you've heard that word before. So we can see that here, a current of maybe 150 picoampere after that will shut off the neuron. So we can already know that that we want a little bit more for our model, that we want these green branches to extend a little bit larger, since we said maybe 250 picoampere should still see some oscillatory responses. This is, if this sounds interesting to you, definitely look into OD2 here at Pitt, the course. So some other steps, or after we have a good representation of a single PPN cell, what would be the next steps? So the next step would be to use what we know about the connectivity within these PPN neurons and connect many neurons to each other, where each is described by that single ODE. or each of their membrane voltages, I should say, is described by the ODE, and then also connects them to a population of SNR neurons, which is already an existing model, but similar formulation, just different currents and some other details. And then we can start trying to match it to some experimental data. So what is often recorded is either in slice, so in a dish after a mouse was, or some rodent was put into a Parkinsonian state and then stimulated, they can slice the brain and still record, but obviously some of the connectivity is lost if you're cutting pieces out. So that changes some things. And then also once the model is somewhat fit to slice data, we can also look at in vivo recordings if available. And then also within all this experimental data, pre, during, and post-stimulation are very important periods. So we can look at even in the non-diseased or the diseased state before any stimulation is going on, maybe have some spike trains like we can see up here. So just black if the neuron is exhibiting an action potential, or white if it's not. And we can see if the frequency matches, if some of the characteristics match. And then lastly, since we are trying to explain some kind of plasticity, some kind of long-term changes within the brain, we will look into what is known about plasticity and what details we might need to include, or what mechanisms we need to look into for this type of plasticity, and we might need to add or keep track of some more details. And when trying to fit these models, one of the measures that comes up many times is the coefficient of variation. So that's the ratio of the standard deviation to the mean. So that kind of gives us a feel for the spread of maybe the firing rate, since neurons don't fire very regularly. But we can see maybe how many neurons fire which rates, or within one neuron, what do the firing properties look like. And then another thing we can adjust is once we get to a population level, we don't want each neuron to be identical, but we know, or it is known that some parameters, for example, the conductance vary a slight bit or a tiny bit between neurons. So that's definitely something we can play with. And then as well as noise, there's a lot of noise in the brain, whether that's the neuron itself, which has random firing, or it came from the experimental recordings. That's always something to consider. And then one thing I wanted to mention is this paper that came out this year, I guess, which has some very interesting ways to analyze the quality or qualitatively compare firing properties. So often an experimental recording or even a model will spit out something that looks like this, as I said, like black and white with action potentials or no action potentials. And how do you measure whether this almost looks like a barcode, but we call them spike trains, is similar to a different one, or is similar to either a different neuron or experimental recordings? So one of the ways we can do that is look at a little window, a subset, draw a Gaussian curve around each spike, and then sum them up. And the width of these can be changed. But then if we sum them up, we have a spike density function and can say at what time along the recording or along the simulation did the neuron exhibit which firing rate. So they're not very regular. Or we could also look at the time between spikes and then use a sliding window to average over those as a different measure of the firing rate. So having two measures to compare data is always better than one. And then this would be an example of a simulation. So we see 10 different trials, could be 10 different neurons, or just 10 different trials of the same neuron. Pre-stimulation, we see different behaviors. And then during the stimulation, some become less active, some first become less active and then become more active. So these kinds of toolboxes and algorithms are very helpful in characterizing the responses of different neurons. And so lastly, I want to acknowledge, well, this was work with my advisor, but also the postdoc, Don Parker, who's been very helpful, and then Erin Giddis and her lab at Carnegie Mellon. And then I know that this Emma Gao is recording currently for data related to this, or that could be related to this project. And yeah, this is still fairly early on, but yeah, thank you very much for their contributions. And then here are some of the references to this presentation.
Thank you so much for the talk. If anyone have question, you can ask.